Okay, welcome, my dear friends. Today's our eminent resource person, Dr.P.Manjula Madam Garu, Assistant Professor, Department of Botany, Government, Rbvrr Women's College, Autonomous, Hyderabad. So, our wish, Madam is very soon is going to join with us, like in regular mode in Government Degree College also. We pray the God and her huge experience and the knowledge, and in previous 2 years and last 2 years onwards, Madam has given her valuable time for the aspirants, those who are already joined in various universities in PG, MSc Botany or MSc Biotechnology in different universities in Telangana and central universities. So none of the other than Dr. P. Manjula Madam Garu. We would like to invite today's our resource person. Madam, please welcome and share your screen I have given to you and start the session, Madam. Please welcome.
Thank you, sir. Sir, is it visible screen? Sir.
It is visible, Madam.
Okay, sir. So today's topic is internal structure of leaf and types of stomata. So simple topic, but it is also important. So internal structure of leaf. So generally, leaf, it contains the following layers. So outer epidermis, so it is a single layered epidermis. So on the epidermis, here the waxy cuticle is present. So here the internal structure of leaf consists of the following layers. First one is a cuticle, so which is present on the upper epidermis, and it is followed by the polisade mesophyll, and next it is followed by the spongy mesophyll. So mesophyll layer, so it is differentiated into polisade and spongy. So next one is lower epidermis. So this is the internal structure of the leaf. So on the upper epidermis and here on the lower epidermis also, so waxy layer is present. So in between the upper and lower epidermis, the layer it is called as the mesophyll tissue. So this mesophyll tissue again differentiated into polisade and spongy mesophyll. So first one is the cuticle. So the cuticle, it is the outermost layer which is present on the upper epidermis. So the outer thick waxy covering of the plants and leaves, generally on the stems also. So this cuticle is present. So it is the outer thick waxy covering. So cuticle which protects the plant from the drying out by the reducing water loss. So due to presence of this cuticle, it mainly helps in to reduce the water loss. So and does not let oxygen or carbon dioxide pass through it. So it acts as a protective layer. So next it is followed by the upper epidermis. So here the upper epidermis, which is consist of single layer of cells covered by the cuticle. So here epidermis generally it is made up of single layer and contains, so on the epidermal region, so small pores are present, so which allows gas or water molecules to pass through. So these pores are called as the stomata. So next one is polisade. So epidermis, which is followed by the mesophyll tissue. So here the mesophyll tissue, that is again upper layer, it is called as a polisade, and inner side it is called as a spongy mesophyll. So just beneath the epidermis, so there are column shaped cells. So these are the column shaped cells which contain the chloroplast. They are called as the polisade mesophills. And most of these, generally it mainly these polisade tissue mainly helps in the photosynthesis due to presence of these chloroplasts. So these polisade tissue mainly helps in photosynthesis. So next one is polisade tissue. These are the column shaped. Whereas next one is the second type of mesophyll, that is it is polysaid tissue, which is followed by the spongy mesophyll, so which is composed of closely packed irregular shaped cells. So they are surrounded by air spaces. So here air spaces are present in the spongy mesophyll. So there are fewer chloroplasts because they are under the side of the leaves. So more amount of chloroplast is present in the polysaid mesophyll only, and on the lower side in the spongy, here mainly in spongy mesophyll, here the storage and also the exchange of gases also takes place due to presence of these air spaces. So next one is it is followed by the lower epidermis. So below the spongy mesophyll is the lower epidermis. So here also that is vascular bundles are also present, and here also stomata. So on the epidermal region, so here the small openings on the lower side and on the upper side, these stomata are present. So they mainly helps in exchange of gases. So stomata. So stomata, these are the small pore-like openings that are present on the epidermal region. So singular form is the stoma and plural is stomata. So and each stoma consists of two guard cells. So generally it contains two guard cells. So here it may be kidney shaped are bean shaped, are in general in monocots it is dumbbell shaped. Generally in dicots they are kidney shaped, whereas in monocots they are in guard shape, they are guard shaped cells. So each stoma consists of two guard cells which control the opening and closing of the stomata. So this is the, so here small opening which is surrounded by the two guard cells. These are the guard cells. So they mainly helps in the that is opening and closing of the stomata. So the main function of the stomata is here which allows exchange of gases, that is carbon dioxide and oxygen and water vapor diffuse in and out of the leaf by opening and closing of these stomata. So here when we observe, so while opening, so these guard cells are turgid, and due to which here the opening of the stomata will occurs, and here when the cells are when flaccid, so the stoma will be closed. So structure of the stomata, generally stomata consist of two guard cells. So when there is a high water pressure, so these guard cells will be open, and during the lower water pressure, so the guard cell will be closed. So generally stomata will open during daytime and close during night. So plants keep their stomata open just enough to allow the photosynthesis to take place, but not so much that they lose excessive amount of water. So here opening of the stomata also for a only for just for few time only, so not for totally. So after some time, so the stomata will close. Otherwise, if the stomata is open for a long time, so here the plants will wilting of the plants will takes place due to here more amount of this more amount of this water will be goes away. So what would happen if the stomata were kept open all the time? So if the stomata were kept open all the time, so water loss due to transpiration would be so great that so few plants would able to take enough water. So if the water loss will occur, so due to which here the wilting of the plants will takes place. So what is the time frame of the opening and closing of stomata? So in general, the stomata are open during the daytime. So when the photosynthesis are active, and closed at the night. So however, stomata may be closed even in the bright sunlight. So in high temperature, so under hot or dry conditions in which water conservation is a matter of life and so in death. So due to which, so generally stomata will open during daytime and close during night, but in high temperatures are due to bright sunlight, so here the closing of the stomata occur in the daytime also, so to preserve to conserve the water. So next one is types of stomata. Is it visible? Stomata PPT. Okay, so next topic is, so here only the next topic is types of stomata. So generally stomata which is present in the epidermal region. So here based on the arrangement of these guard cells and subsidiary cells, so here the stomata are classified into different types, mainly 4 types. So here we already know that here a stoma, it is a small minute epidermal opening, so with the following characteristics. So it contains a central 2 kidneys, so small pore. So it is surrounded by 2 kidney shaped similar cells which containing the chloroplast known as the guard cells, and here, and it is followed by varying number of subsidiary cells. So on the guard cells, above the guard cells, here the epidermal cells which are present, they are called as the subsidiary cells covering the guard cells. So next, a stoma, it is made up of a pair of identical guard cells and forming a pore in the center, through which here the gaseous exchange takes place. So here, here based on the arrangement of these guard cells and subsidiary cells, here the stomata are classified into mainly 4 basic types, mainly in the dicotyledon. So these types are distinguished on the basis of the subsidiary cells surrounding the stomata and their arrangement. So here the 4 types are mainly, that is anamocytic type. So generally it is also it is also known as ranunculus type, and it is seen in the ranunculacean members. And second one is anisocytic type. Generally it is seen in cruciferae. And next one is a parasitic type. It is generally seen in rubiaceae members. And diacytic type, it is seen in the Caryophyllaceae members. So first one is the anamocytic or ranunculaceae type. First one is anamocytic or ranunculaceae types. So the surround this one is the guard cells. So here the arrangement of the subsidiary cells. So the surrounding epidermal cells have no special arrangement and they all are similar, and there is no subsidiary cell. So here there is no differentiation here, the subsidiary cells. So all the cells are normal epidermal cells without any modification, like the stomata of the digitalis, which are seen in the other picture. So here this is called as anamocytic. So there is a no differentiation of the subsidiary cells. So they are looking like a epidermal cells only. So this is called as the anamocytic or ranunculaceae type. Second one is anisocytic type. So here, so the stomata is surrounded by 3 or more subsidiary cells. Here 1, 2, 3 subsidiary cells are present, and here okay. So this one here. So here here 3 subsidiary cells are present. So of which 2 are larger in size and 1 is a smaller one. So one of them is distinctly smaller than the other, like the hyoscamus niger. And so this is here in anisocytic type. So here the guard cells are surrounded by 3 subsidiary cells, and these 3 subsidiary cells, 2 are larger and 1 is smaller cell. So next one is the parasitic. So parasitic type are rubiaceae. So here each stomata, it is surrounded by the 2 or more subsidiary cells, and 2 of them have their long axis parallel to the pore, and which means that axis of the subsidiary cells are parallel to the axis of the pore, like that, like the one in senna leaves. So it is in the senna leaves. So here that is parasitic. So the subsidiary cells, so these are the guard cells. So it is surrounded by the subsidiary cells, surrounded by the two subsidiary cells, and these two subsidiary cells also parallel to the guard cells. So it is called as a parasitic. Parasitic means guard cells and subsidiary cells are parallel. So next one is a, so generally it is seen in Rubiaceae members. So next one is a diacitic type or caryophyllaceae. So each stoma, it is surrounded by two subsidiary cells having their long axis perpendicular to the pore, like in the peppermint, menta and piper. So here these are the guard cells. So here the subsidiary cells, two subsidiary cells, they are present perpendicular to the guard cells. So this is called as a diacitic type. So first one here, this first one is anemocytic. So second one is here, these diacitic means that is perpendicular to the guard cells. So next one is a parasitic, these are parallel to the guard cells, and anisocytic. So here two are large and one is smaller, and actinocytic. So another one is. So here many subsidiary cells are present. So types of stomata and examples of these types of stomata. So this is also important for the identification of the drugs also. Generally the drugs which are obtained from the leaf part. So by identifying the type of stomata also we can identify that this is a correct one, or so here the drug, it is also useful for the drug adulteration. So parasitic or rubiaceous are parallel cells, parallel celled stomata. They are seen in coca leaf and senna leaf. So by identifying the type of stomata also it is useful during the drug adulteration for the identification, evaluation of the drugs. So next one is the diacitic. So generally diacitic are caryophyllaceous. It is seen in peppermint and vasica. Next one is anisocytic are cruciferous or unequal celled stomata. It is seen in the belladonna and datura leaves. So anisocytic type of stomata is seen. Next one is anemocytic are ranunculus types are irregular celled stomata, which is seen in digitalis and lobelia. So in this, that is anemocytic type of stomata is seen. So based on the type, so this type of stomata, it is also useful for the identification of plants or taxonomic groups, and also it is also useful in the drug evaluation. So I will stop here. In next class we will continue. Sir. Sir.
Okay, Madam Ji, thank you very much. So today's our resource person, Dr.P.Manjula Madam Garu, has clearly explained about the internal structure of the dicot leaf. So leaf constraint about the polysac tissue and spongy tissue, Madam has clearly explained. So my dear friends, regarding of today's class, if you have any doubts, discuss or clarify your doubts. Any doubt, my dear friends? Madam, I hope there is no doubt, madam. Madam, there is no doubt. So thank you very much, madam. Tomorrow, same time, we will going to meet in our Zoom class. Okay? Thank you. Manjula, madam.
Okay, sir. Thank you, sir.
Right, madam. Thank you.